welcome everybody to the Protist session in Stream C of the meeting. I realise that although there's many people registered for this conference, there may not be a huge number of people who are working in the field of Protist Genomics or may know a great deal about them. And protists, I should introduce, are not an easily definable taxonomic group. In fact, they're a paraphyletic group that spans everything which is not classified as either fungi, plants, or humans. So I have a phylogenetic tree behind me, which is the only slide I have for this meeting, which I've used in my slide deck for many, many years from Sandy Baldorf in 2003, which shows the eukaryotic tree of life. And we heard from the plenary this morning that the purpose of the objective of the Earth Biogenome Project is to sequence all of the genomes of all described eukaryotic species. Well, by far and away, most of those species will be protists, and the described protist species are probably a tiny fraction, probably a few percent or less of the total number of species of protists in the world. So we're going to hear about from talks today from people who've been trying to tackle the problem of protist genomics in various different ways, and the challenge of trying to work on organisms which can be very difficult to culture, to isolate, which are incredibly diverse, have hugely unusual genomes and gene content, and are incredibly diverse. You must remember that although in this phylogenetic tree, humans and fungi are around here, plants are up here, and absolutely everything else is represented by protist groups. And there's a huge amount we can learn from them if we can tackle the technical challenges of working with them. So the first speaker today is Ross Waller from the University of Cambridge. And Ross has been working for many years on eukaryotic cell structures, endosymbiotic pathways, genomics, and many other aspects of biology in alveolates. And he's going to talk about some of the work he's been doing, I think, today on dinoflagellates.
So dinoflagellates possess some of the most unusual nuclei of all eukaryotes. And I want to tell you today about what we're learning about these nuclei and particularly how it's impacting dinoflagellate genomes. But first of all, let me introduce dinoflagellates if you don't know of these organisms already. They're spectacularly beautiful single-celled flagellates amongst the most abundant and diverse protists in the oceans, according to recent ocean surveys. And you can see in the top right-hand corner, they're blooming around coasts, they're blooming in open oceans around the UK, playing a very important role in photosynthesis. Some of them are very important coral symbionts, so essential for those environments. But in fact, about half of dinoflagellates are non-photosynthetic, and some of these are parasites. So the Norway lobster at the bottom, or scampi, is a fishery that's very heavily impacted by some of these parasites. So having very broad and very important ecological functions and roles. So what do we know about dinoflagellate nuclei? And I'm showing you here an optical section of a nucleus, and you can see already it's got quite a striking presentation. These nuclei have got permanently condensed liquid crystalline chromosomes. And in fact, I can remember from my undergraduate phycology courses, looking at live micrographs such as this one at the bottom there, the instructors said that look out for a bag of sausages as a nucleus for a dinoflagellate. It's a good indicative feature of a dinoflagellate. And indeed, the TEM images on the left-hand side show you just how conspicuous these chromosomes are. So these permanently condensed chromosomes, highly ordered structures. You can see a lot of lateral banding in these. And this state persists throughout the cell cycle. This is a permanent state. So again, very unusual conformation of these chromosomes. The biochemistry of these chromosomes, these nuclei, is also highly unusual. They've got very high DNA to protein content, so more like 10:1 versus 1:1, which you'd find in most eukaryotes. These nuclei also lack nucleosomal packaging of their DNA. And in fact, they've got histone genes, but they're either absent or very scarce as proteins, and they're highly divergent as their primary sequence. So all of this is telling us that these are very unusual, untypical nuclei. And this is not an ancestral state, which was the first assumption in the 80s, but rather this is a derived state, because we know that dinoflagellates are related to apicomplexans, and apicomplexans have got, by and large, pretty typical nuclei. So it's some point after this common ancestry that all of these unusual traits and features have evolved in this group. So we're asking these broad questions. What are the biochemical and biophysical properties of these dinochromatin, if I can coin that term, and how the genome architectures and their expression patterns and processes adapted to these unusual states? So first of all, let me tell you about some of the work we're doing on the biochemistry and the biophysical properties. And on the left-hand side, we started with the proteins. So in this protein gel here, I'm showing you acid-soluble proteins extracted from nuclei, and the left-hand lane is from a canonical nucleus from, in fact, Bachinsis, which is the closest relative of dinoflagellates. And you get typically the four histones, the four core histones, and the histone as the dominant basic nucleoproteins. But from the dinocaryon here, the chromatin, the dinochromatin, we're only getting a single band, and this is not a histone. So the question, of course, was, what is this protein that's the dominant nucleoprotein in dinoflagellates? And we identified this protein as a novel protein. We called it dinoflagellate viral nucleoprotein. We don't find this protein or the gene for this protein in any other eukaryotes, or indeed in any other bacteria or any other cells. But rather, the only place we find it is in a group of large DNA viruses. The groups include the mimiviruses and phycodinoviruses, the viruses that have huge genomes with respect to viruses. So it's interesting we've got this link between a eukaryotic nucleoprotein and a protein in these extremely large viruses. So we've got a structural model from NMR data here. We can see from this that there's a highly ordered structural part of the protein and a C-terminal highly disordered tail domain. And again, this is a conserved feature across both the viral proteins and the dinoflagellate proteins. These are smallish proteins. They're highly basic, similar to histones. So some properties of histones, but certainly a novel protein. So in the course of doing the NMR experiments here, we very quickly found out that if you add DVNP protein to DNA, you rapidly get a phase separation. So I'm showing you here liquid droplets here that are forming, simply mixing DVNP and DNA and seen by fluorescence microscopy. So this is already quite reminiscent of these liquid crystalline chromosomes here. The fact that DVNP is causing this liquid phase separated state. We can do single molecule biophysical measurements on DVNP interacting with DNA. So in this case here, I'm showing you two polystyrene beads that are trapped by optical tweezers, and suspended between the tweezers is a single phage DNA molecule, double-stranded DNA molecule, so about 48 kilobases or 14 microns in length. As soon as we add DVNP to this mixture, you see this rapid contraction of that DNA strand. In fact, that was slowed down 10 times. In between 200 and 500 milliseconds, 14 microns of DNA is being compacted. So this is extraordinarily fast. So already we're seeing this unusual property of DVNP. If we look at this with under fluorescence, and here I'm showing you fluorescent protein, not fluorescent DNA. So the DVNP rapidly coats the DNA that we can see with the linear strand of the molecule there. And then you saw those aggregates forming and that rapid contraction of the DNA. In the second experiment, we're using force to keep the beads apart. So now we're seeing aggregates of DVNP tussling in a highly dynamic process, but unable to contract that DNA there. Again, we can measure the forces involved here, and in this case, it's a force distance graph. So as we increase the distance on the bottom axis here between the two beads, it takes about 7 piconewtons of force to decompact DVNP, compacted DNA. Once it gets to its full length there, we can stretch it out a little bit further, and compared to the naked DNA, we see a different property of that stretched DNA. So we're actually getting a changed mechanical property of the DNA with DVNP. Now, compared to nucleosomes, if we were to do the same experiment with nucleosomes, it would take more like 20 piconewtons of force, and you'd see a zigzag pattern here as nucleosomes are being popped off. So again, we're seeing different properties of DVNP in its ability to compact the DNA, and the forces involved, a lot less force in this case. So very quickly, there are clearly some very different processes and properties associated with managing dinoflagellate nuclear genomes compared to canonical nuclei. So the next question is, what impact does this have on the architectures and the expression programs of dinoflagellates? So we only have a reasonably small sample of nuclear genome data and from a taxonomically restricted group of cells, but we're already getting some good insights from these. From this, we know that and other data, we know that these genomes are huge. The small ones are about a gigabase, the big ones are more like a quarter of a terabase. So these are much orders of magnitude, many orders, so 1 to 2 orders of magnitude larger than their nearest relatives. This corresponds to very high gene numbers, but this is in the context of small exons. So the exons are usually only about 100 to 200 base pairs. Where we do see an increase in size is the introns. So there are lots of introns, and they scale, in this case, according to the genome size. And in the case of hematodidium that we're working on, these introns are then typically tens of kilobases in length. So these genes become very large. With respect to expression, we've known for a long time now that there's very little transcriptional expression or regulation of these genes. So it's presumably mostly post-translational expression control. We also know from these genomic data now that genes are typically arranged in unidirectional arrays. We also know that the messenger RNAs have a 5' transplice leader added to them. And these are properties that are quite often associated in other eukaryotes with some polycistronic transcription, although that's yet to be really fully investigated or defined in dinoflagellates. But we've been doing some high-C analysis, and there's actually some very nice studies just on cyclobutadienase that have been recently bioarchive. And what has been shown here is that these blocks of divergent unidirectional arrays of genes fit within structural domains in these chromosomes that interact very little with each other. So again, this is suggesting that we've got some correlation with this highly ordered pattern in the chromosomes where there's perhaps interaction within these layers, but very little interaction between these layers. And I think, again, this is telling us that understanding the organization of this chromatin and the properties of this chromatin is highly appropriate or necessary for really interpreting and understanding the organization and the expression of these genomes, where we're seeing correlation of genome organization and expression with the architecture of these chromosomes. So finally, I just want to thank all the people that have been involved in this work, highlighted in yellow, including some wonderful collaborators and, of course, funders that have made this work possible. Thanks for your time, and I look forward to answering some questions.
Okay, thanks very much, Ross. So I've got a couple of questions here. First of all, from Fringy. Given the huge range in genome size, what is the median genome size? Are they all 250 gigabase, or is the median 10? So do you have an idea of the range that you're getting from that?
Actually, look, that's a good question, and I don't think it is something which we have a good handle on. A lot of the genome sizes were estimated, quite early on, several decades ago, last century, and even those have been proven somewhat inaccurate. And I think, probably because of the condensed state, the techniques that have been used in the past to try to estimate the size based on fluorescence, by and large, have not always been very accurate. But no, it's a good question, actually. I don't have an answer to that in terms of where we'd expect the median to be, but it's not atypical for them to be very large. In fact, a lot of the effort that has been undertaken to sequence these genomes has selectively chosen the smaller ones. And perhaps the symbionts, like the ones that are in corals and other parasites, often tend to be smaller, and that's probably a secondary reduction in those symbionts, which is something which we see in parasites also.
Okay, thank you very much. Another question is, can you say anything about the methylation patterns in dinoflagellate chromosomes?
Well, we know a little about this, and again, I think we don't have broad enough data to make sort of really good general statements. Dinoflagellates for a long time have been known to have some more unusual methylations or methylation events. So 5-hydroxyuracil is one of them, and but again, the frequency of that modification seems to vary a lot in the measurements that have been made in the past. I think it's just too early at the moment for us to have a good handle on those events and what they might mean in these genomes.
This is I think quite an interesting idea. Someone anonymous has asked, is DVMP being developed as a DNA storage product for use in biotechnology?
So we and I know others have thought about that. We in fact thought at one stage it might be a good mechanism for delivering DNA to organisms. So we tried to see if it would enhance transformation efficiencies, and we didn't see any positive effect in that case. As I say, I don't know whether anybody else has hopped on this from a DNA storage perspective, but it's certainly something which I think we could think about.
Yeah. I'll ask one more question because we are running out of time. Can you comment on the telomere structure? Due to DVMP, is there a canonical telomere structure? Is there something unique? I mean, I know that they're kind of unusual in the apicomplexans, but what about the dinoflagellates?
Well, we know, I mean, telomeres are there, and I think as best as we know that they're relatively canonical. But again, I think we only have superficial insight into the biology of these chromosomes in general. The telomeres from a sequence perspective are pretty canonical, but beyond that, it's still an open question. Okay.
Yeah, so apologies, there are other questions there which you might be able to reply to in chat, or I hope. But thank you ever so much, Ross, for doing this on your birthday as well, and on behalf of everyone at the conference, happy birthday, and thanks for joining us. And that was a really, really fascinating talk. So next in this session is Elizabeth Fernandez from the University of Oxford. Elizabeth is working with the Darwin Tree of Life program at the moment, sampling the protists, but she'll be talking about some work that she did before then, looking at relationship with virus and ocean algae. And Elizabeth, I could just say, has been tirelessly working on Darwin Tree of Life, including moving a microscope to her home kitchen to actually do sampling during lockdown, during the coronavirus, which we were all very grateful for. So now over to Ellie.
Good afternoon. First of all, I would like to thank Neil Hall and the organizers to give me the opportunity to give a talk in this conference. Today, I'm going to talk about a single-cell cell atlas approach we are developing in order to understand host-parasite interactions in the ocean. In the marine environment, no planktonic organisms exist in isolation. Rather, all organisms are involved in a complex network of interactions, which is understood as the marine food web. The outcome of all these interactions determine the transfer of energy across trophic levels, influencing biogeochemical cycles and determining the productivity of marine systems. In this scheme, you can see the classical food web with the main compartments and the main processes. Historically, predation and grazing were considered to be the interactions dominating the marine planktonic food web linkages, whereas parasitism has been largely overlooked. However, with recent advances in molecular tools, environmental studies reveal that parasitism is a widespread interaction in marine planktonic systems and place parasites as key components of the marine food web. So the phytoplankton compartment, which is composed by the primary producers of the ocean, are also infected by parasites. These parasites might be playing the same role than viruses, controlling population dynamics and influencing biogeochemical cycles by altering carbon fluxes across trophic levels. Thus, they might be playing a key role in ecosystem functioning. Yet, the functional nature of such interactions remains a black box, with the possibility of capturing the majority of these sophisticated ecological interactions in culture almost impossible. Consequently, there is little knowledge regarding the biology of microbial parasitic interactions in the ocean, with the molecular mechanisms underlying the infection processes universally underexplored. The application of high-throughput bulk RNA sequencing has been used to obtain whole gene expression profiling of complex microbial communities. However, this approach averages gene expression levels across many cells, whereas parasitic infection stages are variable among single cells. Recent progress in single-cell analysis and the accompanying computational methodologies is allowing to characterize individual cells and their spatial organization on the basis of their RNA expression profile, allowing the classification of cell types and a comprehensive description of the dynamic phenotypes. The analysis and interpretation of this data has allowed to capture the cellular heterogeneity in a high-dimensional landscape, which has revolutionized our understanding of tissue development and illness. However, this approach has been mainly applied to animal cells. An example of this approach is the Human Cell Atlas Consortium, which aims to define all human cell types in terms of their distinctive patterns of gene expression, physiological states, developmental trajectories, and spatial relationships in tissues. Another example of the application of these methods in a human disease caused by a protist parasite is the malaria cell atlas. This approach has allowed high-resolution mapping of developmental processes, cellular diversity, and cell-to-cell variation between parasites across their full life cycle. So these cell atlas approaches have placed cell-cell interactions at the vanguard of marine microbial research. Very recently, the application of single-cell RNA-seq in a marine virus phytoplankton system has shown it is possible to track host pathogen infection dynamics at high resolution. Clustering cells based on their similar expression profiles through time allow to map the infection progression across a continuum of infection states, and also to divide viral genes into kinetic classes across the infection stages. However, further development of this approach is possible if omic methods can be combined with high-throughput cellular imaging, so that the progression of a cellular interaction versus transcriptome state can be quantized. So the combination of these methods will allow us to deconvolute cell-cell interactions occurring in highly complex systems, as it is the marine environment. So the development of this approach will be a huge step forward in the study of the complex microbial interactions that occur in the ocean. To overcome this limitation, we are currently developing a single-cell cell atlas approach in a marine protist single-cell parasite host association, using Perkinsea parasites of toxic bloom-forming dinoflagellates as a model from culture and from the environment. So dinoflagellates cause harmful algal blooms in coastal areas worldwide, which have negative consequences for the ecosystem, human health due to the production of toxins, and affect local economies by impacting aquaculture and tourism. During the past decade, several studies highlighted these blooms co-occur with protist parasites. These parasites present a similar life cycle with different life cycle stages, as you can see in the scheme at the bottom left of the slide. In brief, a zoospore penetrates inside the host cell, consumes the host cytoplasm, and then divides, producing new zoospores that are released into the environment in order to seek a new host to infect. So in the natural environment, we can find at the same time all stages of the life cycle. This is why it is so important to be able to capture all the variation in the gene expression profile at the level of single cells and relate them to the cell states. For instance, in the pictures, you can see circled in blue a healthy host cell. We also can see early feeding stages of infection, late feeding stages of infection, also stages when the parasite starts to divide and form the zoospores, late stages of infection ready to release the infective zoospores, and also living zoospores seeking a new host to infect. So ecologically, these parasites are important because by killing these blooming cells are contributing to bloom termination and nutrient cycling. The Perkinsea parasites that comprise our model system belong to the phylum of alveolates, together with dinoflagellates, ciliates, and apicomplexans. The knowledge of the biology and diversity of this group is still very scarce. So most of its diversity comes from environmental sequences, with only a few species properly described, and all the described species are known to be parasitic. Also, there is not much genomic information about them. So we have some of these Perkinsea parasites in culture in our lab. So our approach consists of sampling a co-culture of Parvulocifera alexandrium system through time during the whole infection cycle. We sort single cells using fluorescent activated single cell sorting into 384 well plates, which later we image them with a high content imaging system. Since the host cells are three-dimensional, we are taking Z-stack images. That is allowing us to detect early stages of infection that are located in different positions of the host cell. We also are currently developing an algorithm for the automated classification of the single-cell images into cell states. This classification is based on the combination of cybergreen and chlorophyll fluorescence. At the same time, after taking the images, we will carry out single-cell RNA-seq in order to obtain the transcriptomic profile of each of the single cells. The combination of single-cell RNA-seq with high-throughput cellular imaging will allow us to quantify the progression of a cellular interaction versus transcriptome state, giving us a better resolution of the continuum of the infection cycle and the deconvolution of the interaction. So to show you some numbers, starting from a culture of only 30 milliliters, which is a very small volume, with a concentration of 10 to the 4 host cells per milliliter and a ratio of zoospores versus host of 20:1, the application of this approach has given us thousands of sorted single cells across the full life cycle that are confidently classified into a continuum of cell states. At the same time, carrying out single-cell RNA-seq is allowing us to obtain thousands of transcriptomic profiles associated with each image, and therefore to cell states. After applying this approach in culture, we will do the same process with 2 other dinoflagellate parasite species of Perkinsea belonging to a different genus. This will allow us to check how applicable is our method to other Protist host parasite systems. And finally, we will process an environmental sample during a dinoflagellate bloom event, comprising different parasite species and all ranges of infection states. So in conclusion, the development of a single-cell atlas approach to marine Protist host parasite systems will transform the way we study single-cell interactions and the way we understand cellular systems that determine critical interactions shaping marine environments. To finish, I would like to thank the people in Exeter who are developing the automated image classification algorithm, the undergraduate student who is working on the images and helping in training the algorithm as well, people in the Marine Science Institute for providing parasite host co-cultures, and also the other members of Richard's lab. And thank you for your attention.
Okay. Thank you very much. That's an excellent talk. I just have a question straight off. There aren't any in the chat at the moment, but there may be some that turn up. Do you have any idea about how, are they species specific, the Perkinsea parasites, or do they infect a range of dinoflagellates? And are there other species which infect other species of dinoflagellate?
So as far as we know, all the studies have tested the whole range in the lab, and all the parasites that have been described that infect dinoflagellates are generalist parasites, so they can infect a wide range of dinoflagellates. However, when you look into the field data, what we can see is that most of the time, it seems that blooms produced by one, or that are dominated by one dinoflagellate species, there is also one parasite that it's predominant with this species, but it's like very little known still about how specific or how specificity works in these parasites. Okay.
And we have a technical question there. What kind of coverage do you get from a single cell after ribosomal depletion of the transcriptome?
So this is a work that we're still developing. So we are not, we don't have the transcriptome data yet. So we are still in the process of getting the images and training the algorithm to classify these images. So then this is a work that we are developing at the moment. It's not a work that we have results yet. Which is the coverage of one single cell.
Yeah, and one other question is, if with the transcriptome, so you might not have an answer yet, but you're going to get a combination of host and parasite. So do you have references for these genomes, and if not, how will you deconvolute the two?
So we have some results from transcriptome data of the parasite.
Yeah.
And what we saw is that the parasites have a different splice leader sequence than the host. So one way to separate this is to use the splice leader in silico to separate transcripts from the host or the parasite. And then we also have genomes, 3 genomes of these 3 different parasites available now. And we also have transcriptomic data. So another option would be like to map the rates to this data set, reference data set.
Okay, right, Ellie, thank you very much for an excellent talk. That's very cool work. So the next speaker in this session will be Sally Waring from the Earlham Institute. Sally's previously worked at the Natural History Museum in New York and NYU, and she's working in the Darwin Tree of Life program and developing the tools to try and sequence protists en masse directly from the environment. So Sally, over to you.
Hi everyone, I'm Sally. I am a postdoctoral scientist at the Earlham Institute, and I'm going to be talking about why we need more protist genomes and what we're doing to create more protist genomes as part of the Darwin Tree of Life Eukaryotic Microbes project. All right, so this is a figure from a review in 2014, and what I want you to take away from that is that protists are underrepresented in genomic databases. So we've got these pie charts here with the number of described species, total 18S rDNA sequences in databases, total number of different 18S rDNA sequences from environmental samples, and the number of genomes available in databases. And we've got these different groups of eukaryotes colored differently here, and the protists are in yellow. And the takeaway message from here is that we have very few described protist species among the eukaryotes. We have many, many environmental 18S rDNA sequences, indicating that there are many undescribed species of protists out there. And yet we have very few, or comparatively few genomes for protists compared to some of these other groups of life. And if we go into a little bit more detail on the groups of protists that we have genomic information for, this is a review from 2017. What we have here are kind of major groups of eukaryotes in this tree, and then these bar charts around the edge showing counts of number of genomes and transcriptomes that are available in various databases. And what we can see from this is that a lot of these genomes and transcriptomes are concentrated in particular branches on this tree, right? So things like the apicomplexer, which include important protists and parasites, have lots of genomes, as do the land plants and green algae, and animals and fungi here are well represented. But a lot of these other branches that represent protist species that are less studied have few or no genomes or transcriptomes at all. There's over here things called orphan lineages, which are protists that have not really, they don't have a determined place yet on the tree of life, or they're not really part of another one of these lineages. And these are particularly poorly represented when it comes to genomic information. All right, so why is it that protists are underrepresented in these genomic databases? Well, it's often because protists are hard to isolate and hard to identify. So what I've got here is a video of just a drop of water taken from a freshwater pond. It's from Brooklyn in New York. And what I want you to see here is that this one drop of water from this pond is full of different protist species, right? So we've got these little cells moving around. There was a ciliate that came through there. These little algae here are cryptophytes. And then there's a little algae here. There's all these other shapes, these long kind of thin shapes, which are various types of diatoms and green algae. There's other funny little cells. That's another ciliate there, a colep ciliate. There's a tiny little cell there. I don't even know what that is. And when you're a protistologist and you go to these environments in the wild and you collect water samples, you very quickly see that it's very rare that you collect a water sample in which you don't see something which you can't identify, right? And the traditional way of identifying and classifying these things is to take a water sample like this, physically isolate some of these cells, get them to culture in the laboratory and do detailed analyses of their structure, their cell types, and of their genomes as well. And that's a fantastic way to properly describe these different organisms and get their genomes, but it's incredibly labor intensive and it's difficult. Some of these things don't lend themselves well to culture, particularly, some of them need various bacteria and other environmental conditions that we don't really understand. And so it could be very difficult to both describe these species and generate genomes for them. All right, so as part of the Darwin Tree of Life, we wanted to develop additional ways to both describe protists that we found in the wild using genomic technologies and to create more genomic genomes for protists, right? So I'm just going to take you through one of our projects was the single cell sequencing using G&T-seq, which is where you sequence both the genome and the transcriptome from single cells. This is something we're working on at the Earlham Institute and with Oxford University as well. So we start with an environmental sample, filtered water sample, which contains a mixture of cells. We're planning to do community DNA extraction from those samples. So, and we want to, from those community DNA extractions, we want to generate long reads and possibly high-C data as well. And the other thing that we're doing is taking part of that sample and sorting it into a plate. So when we do that, we get all these individual cells split out into these individual wells on a plate. Then we can take those individual cells through a G&T-seq protocol where we get whole genome amplification and short read sequencing, and also whole transcriptome amplification and sequencing. What we plan to do eventually is kind of combine these data sets so we can bin this really useful long read data using our short read data from these single cells and to get nice kind of hybrid data sets, and then eventually join all that together into an assembly, incorporating high-C data and using this transcriptome RNA-seq data for annotation as well. So today I'm just going to show you just a tiny amount of data that we've got so far, just really focusing on this part of the protocol, the G&T-seq from the single sorted cells. Right, so we started off just with one pilot plate, just to kind of as a proof of concept to see whether we could do this with these different cell types. And this, because we were originally planning to obviously use a cell sorter to sort these cells, but because of lockdown, I want to highlight that Ali Fernandez here from Oxford University took a microscope home with her and manually sorted these cells into individual wells from environmental samples. So she goes through those environmental samples, physically picks out these cells and puts them into a well on a plate. So this is just some of the pictures of some of the organisms that we managed to isolate. Some of them are quite distinctive. This one here in AL11 is a green alga called Pediastrum. We have a nice euglena here, a ficus. And then you have other things that are not quite so distinctive, like this little blob here, or this one from A6, which is my favorite, which is just this tiny little cell here, just kind of zooming off in the corner of the photo frame. All right, so we did a very low-level kind of skim sequencing of both the whole genome amplified DNA and the transcriptome amplified cDNA from this first pilot plate. This is just a graph showing you kind of the number of reads, so it was low coverage, 100,000 reads or read pairs total. And then we've got cDNA in pink and gDNA in blue. And the different kind of cell types are colored down the bottom here. We had a few controls, including Chlamydomonas, human cells, and yeast. And our first kind of thing we want to know is just, do we get data from these cells? And the good thing is that we do. So we managed to generate both cDNA and gDNA from most of these samples, not all of these samples. Sometimes we have problems, but we do manage to get reads from both. And then the other thing we're able to do with the skim sequencing data is do some taxonomic classification. So we've tried many ways to do this. I'm just showing you the result of one of the ways we did this, which was using centrifuge to do taxonomic classification of the cDNA reads. And this plot here just shows you the percent of reads that turned out to be of eukaryotic origin. So we can see we get variable amounts through this plate here. And we also, in this top row here, I'm showing the kind of the eukaryotic group where most of the reads fall in this classification protocol. And something like, these first two here are actually our control Chlamydomonas cells, and they're coming out, most of the reads are falling into the verteplanta lineage, which is great. And something like A7, which is an unknown chlorophyte. And again, that's coming out mostly as a verteplanta. And then next one here is a dinoflagellate, and that's coming out as most of the reads are alveolata. So this is really great for us. We're like, okay, we're getting reads, and it seems like some of them are coming from the cells that we want them to come from. It's not always the case, right? A12 here has very few eukaryotic reads, and that's because most of the reads are getting sucked up by bacteria, which is a problem with a lot of these environmental samples. They can be full of bacterial genes or DNA. All right, so from this, we picked a few cells, ones that we thought were either that showed kind of nice data, so lots of eukaryotic reads and a good number of reads, and were things that we're particularly interested in. And with those, it's 5 cells, we're doing deeper sequencing. So we kind of guesstimated the genome size of these organisms just based on other available genomes, and we aimed for about at least 100x coverage of all of these genomes, and we went back into those cDNA and gDNA samples and sequencing them at more coverage. So we've got things like diatoms, a euglena, a dinoflagellate, and this little unknown one here. All right, so that's stuff in progress. We don't have any results to show at this point. So what are we planning to do? So we're doing this deep sequencing. We want to use that to determine what the level of coverage we're able to get from this kind of protocol, and what we kind of need to get decent genomic data, and also what's the completeness that we're able to get from this kind of project, and how much genomic information can we get out of this GT-seq protocol. We're also moving on to automated cell sorting eventually, and we think we'll be able to do cool things like tailoring the cell sorting to pull out cells with particular characteristics, such as heterotrophic flagellates, which are often undersampled in these environments because there's just so many of them and they're so small and they're hard to work with. And we've got additional plates to analyze using different cell types, different cell numbers. We're experimenting with, is it good to have maybe tens of cells rather than one cell? So if we leave the plates for a little bit of time and allow the cells to grow a little bit, does that give us better data? And all this is going into a pipeline. So we're doing all these genomes, we're doing all this analysis using a pipeline rather than processing everything manually. And I wanted to just highlight that the postdoc that I work with at the Yale Institute, Jamie, is responsible for that pipeline, is doing fantastic work getting this pipeline up and running so that we can process all this data really quickly. And just as a summary of the outputs of this project, what we want to do. Okay, so we want more genomes covering more species from various branches on the eukaryotic tree. And we want to use these single amplified genomes to give us hopefully a better idea of who is there in these environmental samples and how many different taxa there might be. And also we're hoping we might get some metabolic information. So if we can annotate some genes, get an idea of what everyone's doing, who's eating who, and all that kind of stuff. And develop pipelines and protocols for future studies. So thank you very much for listening. There's lots of people involved in this project, lots of people at the Yale Institute, particularly Jamie, who is the other postdoc on this project, who's doing a lot of the pipelining, and the University of Oxford as well has a team who we are working with to do this. So thanks to everyone who is involved, and thank you for listening.
Okay, thank you very much. It was really cool stuff. I would say that, I guess. So we have some questions in the chat for Sally. So first of all, so most of the Protist data that's there at the moment, is it from freshwater or marine environments?
Yeah, I actually don't have numbers on that, but typically freshwater environments are understudied in comparison to marine environments for Protist diversity. So I would predict that there are more marine species in the genome databases, but I'm actually not 100% sure. Okay, and a highly scoring question. Let's move to the top. Is there a strategy to enrich the genomes for underrepresented clades in the tree that you showed at the beginning there? Yes, so some of those, not so much on the genome level, but on some of the process level. I mentioned, we can use different stains to try and target cell types that we like. So a lot of the underrepresented lineages are heterotrophic flagellates, right? So we can do things to exclude photosynthetic cells and look for the presence of flagella on our cells, and that can help us to increase the number of those types of cells in our plates.
And so there's a question here about, do you obtain genome size estimates from the initial sequencing? Otherwise, how do you know you obtained 100x coverage from single cell Illumina or PacBio?
Yeah, that's a total estimation. So we don't have, for the data I showed you, we don't have good idea of the genome sizes of those particular organisms. So what we did for those ones was we looked at other genomes from as closely related lineages as we could find, and thought, okay, well, genomes from this lineage tend to be in the range of tens of megabases or hundreds of megabases or gigabases, and that's what we went with. But from the small amount of data that we got from the skin sequencing, we're not able to predict genome size because we don't have enough coverage.
Okay, so one other question: do you think that the bacteria you got in the cDNA and gDNA might associate with proteobacteria, for instance? So I guess do you have an idea? Are you looking at where the bacterial contamination is coming from? Is that of interest?
There are definitely a lot of proteobacteria in there. Some of these data sets are coming up with sort of, it really depends on each well can be a little bit different, but some of our wells are coming up as sort of majority of the bacteria, protobacteria.
Okay. Yeah. Great. So I think in the interest of time, we'll have to move on. You've got a few more questions that we have time for that hopefully you might be able to respond to in the chat. But thank you very much, Sally. That's really interesting work. And now we're moving on to Adam Reeves, the final talk in this session. Adam is at the Sanger Institute working with the parasitology team there. And he's going to give a talk on a really, really cool and interesting parasite, which I've kind of been interested in these for a while. Never worked on them, unfortunately. Hepatocystis, which is related to the malaria genomes, and from data they mined from the red Columbus monkey genome. So Adam, over to you.
Hi, my name's Adam Mead. I'm a senior staff scientist at the Wellcome Sanger Institute, and I'm going to talk to you about our hepatocystis genome and what it can tell us about malaria parasites. So a colleague of ours, Theo Sanderson, who used to be at the Sanger, he's now at the Crick Institute in London, came across some serendipitous sequence. He was searching with a plasmodium gene, and he found what looked like some very plasmodium-like sequence in a red Columbus monkey assembly. It's the red Columbus monkey on the right-hand side here. So this was very strange, and when he looked more closely, he found that actually a lot of the contigs in the Columbus monkey assembly matched Plasmodium vivax, one of the malaria parasites, and they had a distinctively low GC content compared to those that matched monkeys. So it looked like the blood sample from this monkey contained quite a lot of parasite material. This had been sequenced. And when he took a particular gene from that assembly and drew a phylogenetic tree, he found that the sequence clustered with hepatocystis, which is a genus that is very closely related to Plasmodium parasites and known to infect these kind of monkeys. So let's find out a bit more about hepatocystis. So it's an epicomplexan parasite, a protist, closely related to malaria, infects the liver and red blood cells, and it's known for visible cysts in liver tissue, hence the name. It infects monkeys, bats and squirrels. It's thought to descend from malaria parasites, and the reason it's not called Plasmodium but has its own genus is that it doesn't have some of the key features of Plasmodium parasites, one of which is it's thought not to replicate in the blood. And instead of using mosquitoes as a vector, it uses biting midges, Culicoides midges. So if indeed this parasite is descended from malaria parasites and doesn't replicate in the blood, can we use its genome sequence to learn something about how malaria parasites replicate in the blood? So that's the question that we posed. So first of all, how good a genome assembly could we get? Well, if we just took hepatocystis contigs from the original monkey assembly, there were about 12,000 and the M50 was about 2.4 kb, which isn't very good. So Eric Aunin from our team spent a good deal of time coming up with a bespoke assembly pipeline for this data, and he managed to get the assembly, it's a short read Illumina assembly from a monkey blood sample down to about 2,500 contigs and at 18.4 kb, which is quite kbn50, which is quite respectable. And we found over 5,000 genes, well in the kind of right range for Plasmodium type parasites. And when we drew a nuclear phylogenetic tree based on lots of thousands of orthologs conserved across Hepatocystis and Plasmodium, it was clear that Hepatocystis is an outgroup to the rodent malaria parasites here, and embedded right within the Plasmodium genus. And previous phylogenetic work using small numbers of genes from the nuclear genome, the mitochondrial and apicoplast genomes was not very clear. And we've shown that the reason that it wasn't very clear is that the nuclear and mitochondrial and apicoplast genomes don't agree very well with each other. But thousands of genes on the nuclear genome clearly place Hepatocystis here. So we wanted to see if we could find any evidence for schizonts in the blood, see whether these parasites might actually be replicating in the blood. The dogma really is that they don't, but there has been recent evidence that maybe the schizonts were maybe found in monkey blood. So to do this, we were fortunate enough to actually have 26 RNA-seq samples from the group of monkeys, such as the one that we got the DNA sample from. And Eric found that all these RNA-seq samples showed lots of parasite material, and all these parasites are very similar to each other. So a whole group of these monkeys were all infected with essentially the same parasite, quite high parasitemias. And we used a deconvolution approach for this bulk RNA-seq data using the recent malaria cell atlas data that we were involved in producing. So we could figure out what selection of different life cycle stages of malaria type parasites were in these samples. And there was no evidence whatsoever for schizonts, just early ring and trophozoite blood stages, and then these mature transmissible forms, the male and female gametocytes. So then we went on to try and identify genes that might have been lost in Hepatocystis related to this bit of the biology that seems to be lost. And the way we did this was again using the malaria cell atlas. And with that data, all the genes in Plasmodium were clustered, so to find those which follow the same pattern of expression across the life cycle, from the liver to the blood to the mosquito. And we looked in each of these clusters to find where more genes were missing in Hepatocystis than you'd expect by chance, given that they're conserved across Plasmodium. And we found that the only cluster with a significant number of missing orthologs was cluster 10, where the genes tend to be expressed highly in the late schizont stage, which is exactly the bit that seems to be missing in Hepatocystis. And this comprised all of the reticulocyte binding proteins, which are known to be involved in red cell invasion. And independently, we noticed that there are almost no peer genes, which are the largest multi-gene family in Plasmodium and are thought to be involved in maintaining long-term chronic infections in the blood. So what does this mean for our understanding of malaria biology? Well, one of the most intensively studied parts of the malaria life cycle is this erythrocytic schizogony that's lost in Hepatocystis, where the parasites come out of the liver, invade red cells, replicate, and then go on round and round invading red blood cells. And the reason is this causes the symptoms of malaria, so the anemia and the fevers. And if we can block the parasites from invading red blood cells, then we can block both, we can prevent the disease and also prevent transmission of the parasites to the mosquito. And what our data is telling us is that malaria-type parasites can invade red blood cells straight out of the liver without reticulocyte binding proteins, which we know are essential for these subsequent invasion steps. So it suggests that the invasion pathways used early on may be different from those later on, and this is an underappreciated area of this biology. Another interesting point is that where in malaria parasites, most parasites will come out of the liver, invade the red blood cells, and then continue to go around invading red blood cells. And only occasionally will they become committed to form gametocytes, which are the transmissible stages. Whereas in Hepatocystis, they come out of the liver, invade red blood cells, and are immediately programmed to develop into gametocytes. So this switch that happens in Plasmodium parasites is missing or turned on by default in Hepatocystis. And so if we can better understand the genomic basis of this, in Hepatocystis, we can maybe better interfere with transmission. So in conclusion, we used read sequence from whole monkey blood. We assembled and annotated a draft quality genome of Hepatocystis ex Piliaclobus trophosceles, Hepatocystis from the red Clobus monkey. And we confirmed the phylogenetic placement as an outgroup to the rodent infecting Plasmodium species. We found no transcriptome evidence for schizont stages in the blood. And this lack of blood schizogony is supported by missing orthologs often being expressed in Plasmodium blood schizonts, so in particular the red reticulocyte binding proteins and the pear proteins. So what does this tell us about malaria parasites? Well, it highlights the distinction between first and subsequent generation merozoites, which is a really neglected aspect of red cell invasion studies. And we think a better quality genome sequence might give us insights into the control of transmission stage development. And that's something we'd like to work on. So I'd just like to thank particularly Eric and Uli, who did much of the work in our group, and Matt, our head of our lab, Chris Newbold at Oxford, Theo at the Crick Institute, who really led us to this study by finding the sequence, and then the original people who did the study who came on board with us and helped us out with some of the raw data, Tony, Noah, Nelson, and Colin. Thank you very much.
Okay, thank you. Excellent talk. So we have a couple of questions. We've got a few minutes left in this session. We're running really well to time. So first one from Meg Allen, who's asked, have the sequences of this parasite, I guess, been obtained in the vector yet? And can you maintain the vector in the lab?
So as far as I know, we don't have any sequences from the parasite, I guess, obtained from the vector. I'm not familiar actually with whether coulicoides can be maintained in the lab, but I'd be interested to know.
And also there's a question about you mentioned the early invasion pathways you've identified. Are these found in many other populations, or is this specific to this particular case? I guess, do serial invasion events occur? So I don't know if this is part of the same question, but do you know if this is those early invasion pathways are common to other malarias, I guess is the question.
So I guess, so what we haven't done, unfortunately, there is identify what the early invasion pathways are. I guess we've ruled out the, well, we've ruled out the involvement of reticulocyte binding proteins in Hepatocystis. Clearly, they happen later on in Plasmodium parasites, but we haven't been able to say what does happen in Plasmodium parasites in the first generation merozoites, unfortunately.
I see. But so a related question is, does serial invasion occur in the liver? So do liver-derived merozoites reinfect other liver cells? And are there multiple invasion pathways there? I guess is
I don't know. It's a really interesting question, actually, because one of the really fascinating things about Hepatocystis is the infections can last, I think they've been recorded up to 15 years. However, they must be maintained from the liver. And it's not clear that there are hypnozoites or anything like that in the liver. So whether they dormant in the liver or whether they continuously kind of reinvade in the liver, I don't know the answer to that. I read the Genome chapter on it, and I can't remember. I don't think there's enough known about it. It's really quite understudied.
Liver stage is a bit of a mystery, I believe, isn't it? So I have just a quick question from me, really, which is: so you identified missing genes. Do you have any idea about what's happening in terms of gene synteny or rearrangements or deletions that are occurring, or is the genome not in good enough shape to say?
So yeah, the genome's pretty drafty. We haven't seen very much synteny. We've seen regions that look to be like the classic subtelomeric region in most Plasmodium genomes. So where you get a series of the less well conserved genes and multitude families. But we don't have very big contigs, and we'd really like to go back and get some much better sequence.
Okay, so I thank you very much. That was a really great talk and a good one to end on. I should just say that Mark Blackster has put in the chat to remind everyone to go to the poster session, and he's put a link in there at Spatial Chat at BG 2020, so you can continue discussions and view posters in that forum. I just want to thank all of the speakers for a really excellent session and keeping beautifully to time.